Welcome to Chapter 8, Aerobic Metabolism, Part 2 video. You should turn in your PowerPoint packet to pages 111 through 113. Hopefully you've already filled in, using the pages in your textbook and pencil, the answers to these components. If you have not, I would take the time to do so before you listen to this video. The answer key is also located on Blackboard. I would try to complete it first without the answer key and then check your answers. So what we're going to do is walk through aerobic metabolism. So not all bacteria are able to do aerobic metabolism. Some can do anaerobic or fermentation. The majority of bacteria make their ATP by aerobic metabolism, and this is the most complex of the three types of metabolism. Let's look at figure 816 before we begin the pages 111 through 113. This figure from your textbook is comparing cellular respiration to fermentation. So let's talk a little bit about terminology. You probably have talked about aerobic respiration in your other classes. So the word aerobic means with oxygen. The word anaerobic means without. This is also called cellular respiration because it occurs within the cell. It is also called respiration, not because the bacteria is breathing, but because oxygen and carbon dioxide are used and produced. So in this figure on the left-hand side, you can see glucose is the beginning substrate or beginning molecule. If you count the number of gray circles, it is 6. Recall from video 1, glucose is C6H12O6. For ease of understanding, we're going to focus in these tutorials on the number of carbons and how those carbons change throughout the process. So let's just look at the left-hand side of the page. We see glucose, which is a 6-carbon molecule. It's undergoing something called glycolysis. The next step is either called the intermediate step, the prep step, or sometimes called the transition step. They are all the same step. The third step is called the Kreb cycle or citric acid cycle. And then we have what's called the electron transport chain. So that is a brief overview of what we're going to discuss. What you will also see is NADH and FADH2s at the bottom of the screen and ATP being produced. So let's go back and review a little bit about NADH and FADH2s. So NAD plus and FAD are electron carriers. Electrons have a negative charge. So when you see NAD plus and FAD, as you can see here on the right side of the screen, they're what I call empty train cars. They no longer have passengers. And how do I know that? NAD plus and FAD do not have the H or the proton hydrogen in their name. On this side, you see NADH and FADH2. Those are what I call full train cars. They have passengers. What are their passengers? Hydrogen and electrons. Remember, their job is to transport or carry hydrogen and electrons. So on the left-hand side, they have passengers, and on the right-hand side, they are free to pick up electrons and protons. This is important for redox reactions, which we mentioned in the first video. Redox reactions, if you recall, is a series of reactions in which one molecule is oxidized, which means it loses electrons, and one molecule is reduced or it gains electrons. If you see the hydrogen present, that means it's carrying electrons. If the hydrogen is not present, it does not have a gain of electrons. Again, the entire point of metabolism is for the microbe to make ATP. On the right side of the screen, you will see the word fermentation. What you will also see is that you're going to make less ATP, which we'll discuss in the third video. You still start with glucose. Glycolysis is performed. We still see NAD plus and NADH. However, the process is slightly different than an aerobic or anaerobic fermentation. So let's begin with things that you're comfortable with. This is figure 817 from your textbook, where cellular respiration occurs. So we're comparing and contrasting prokaryote cells to eukaryotic cells. So let's go back and review what you have learned in your previous courses. A eukaryotic cell, such as a human cell, a plant cell, a fungal cell, contains a cell wall, perhaps, if you're a fungus or a plant, a cell membrane, and organelles. Organelles like a nucleus, SER, RER, and mitochondria. So in a eukaryotic cell, glycolysis occurred in the cytoplasm, and the remaining of the steps, the intermediate step, the prep step, the Kreb cycle, and the electron transport chain, seen here as 1, 2, 3, and 4, are going to occur either in the cytoplasm or in the mitochondria. However, this course we are discussing prokaryotic cells, specifically bacteria cells. And if you recall, prokaryotic cells do not contain a mitochondria. This is actually a little more simple. So, in a bacteria cell, glycolysis step 1, the intermediate step, step 2, the prep step is the same as the intermediate step, the Krebs cycle step 3, all occur in the cytoplasm. The only step that does not occur in the cytoplasm is step 4, or the electron transport chain. In the electron transport chain, as we will see, electrons need to be shuttled across a membrane. But in this case, it is not a mitochondria, it is the cell or plasma membrane. We will go to each of these steps in detail as we progress through the worksheet and the slides.
So before we begin looking at page 111 in your PowerPoint packet, let's get an idea of the big picture and what's happening here. So on the left we have an uninoculated nutrient agar broth. So basically we have a broth with no bacteria added. We then would use our loop to aseptically transfer 1 or 2 loops to the nutrient agar broth. The result, in this case E. coli, would be a nice turbid or cloudy broth. So what is really happening here? We are adding bacteria to a test tube and allowing the bacteria to have everything it needs to grow. The right temperature, the right salinity, the right pH, the right food. So the food source in this broth would allow the bacteria to divide. Once it does division, it's actually doing metabolism. So the bacteria is using the food in the nutrient broth to make more bacteria cells. So in this picture, we're getting a little bit more specific. So now we have a test tube of E. coli that's already grown, it's been inoculated, and it's allowed to do binary fission. And in the second picture, we have what's called a Durham tube, which is an inverted test tube that is full of media. The media in this picture is red. You've seen a Durham tube before when you did the water experiment with lactose broth. In this picture, we're looking at just a sugar tube. It could be lactose, it could be glucose. Let's assume that it's glucose. So the tube is red, which is called phenol red, and it's going to tell us an indicator of whether or not an acid has been produced. And the Durham tube is full of media. So what does this have to do with aerobic respiration? We're going to use a loop to inoculate this Durham tube with E. coli. And then after 48 hours of incubation, if E. coli has done metabolism, if it has used the glucose in the tube, you're going to see a gas bubble, which is in the Durham tube. You're also going to see a color change, which is from red to yellow. The yellow indicates acid has been produced. This will be explained further when you view the physiological test PowerPoint slides for Lab 10. Now let's go to the worksheet, page 111, and look at the images. So this slide shows a summary of aerobic respiration, specifically in prokaryotes. So before we begin looking at pages 111 through 114, let's look at an overview of what we're going to do. So we start off with step 1, which is called glycolysis. Glycolysis is the split or breaking down of glucose. Glucose is a 6-carbon sugar. During glycolysis, which requires 10 steps, glucose is split into 2-pyruvic acid. During that process, which occurs in the cytoplasm, 2 ATP are produced. 2 NADHs are produced. In the second step, which is called the prep step or transition step or intermediate step, pyruvic acid is then broken down into something called acetyl. So what has happened is glucose, which has 6 carbons, is split in half. 6 divided by 2 is 3. Each pyruvic acid has 3 carbons. The pyruvic acid then loses a carbon, giving us a 2-carbon molecule, which is acetyl. During the prep step, which also occurs in the cytoplasm, 2 NADHs are formed and 0 ATP. When each of the carbons that is lost during pyruvic acid is going to combine with oxygen to form carbon dioxide. Next, the acetyls combine with something called coenzyme A to form acetyl-CoA. Acetyl-CoA is going to join the Kreb cycle or citric acid cycle. This also occurs in the cytoplasm. As the Kreb cycle spins, 4 carbon dioxide are formed, giving us a total of 6, 2 from the prep step and 2 from the Kreb cycle. The citric acid cycle or Kreb cycle also produces the majority of the NADH and FADH2. Recall that NADH and FADH2's job is to carry electrons and protons. During the Kreb cycle, 2 ATP are produced. The last step, called the electron transport chain, produces the majority of the ATP. This step is going to transport hydrogens and electrons across the cell membrane in prokaryotic cells and across the mitochondria of eukaryotic cells. The NADHs and FADH2s from the previous step enter the electron transport chain and then are shuttled across the membrane to form what's called a concentration gradient. As that gradient moves and electrons move, ATP is produced. During the electron transport chain, the last step of the process, 34 ATP are produced for a total of 38. Also, 6 water are produced. This is a brief summary or overview. We will go through each step individually, look at the worksheet in the PowerPoint packet, and look at animations as we explain each step. We're now on page 111 in your PowerPoint packet. Follow along with this particular page. Hopefully, you were able to successfully fill in with pencil the correct answers. The answer key is also located on Blackboard. It is to your advantage to do the answers first and then check the answers afterwards. So let's begin with the picture. So we have at the top the words respiration and fermentation. We're going to follow the path of both of these for different bacteria or different microbes. So in respiration, we're going to use oxygen, and in fermentation, we are probably not going to use oxygen. So we're going to start with the first box. It says, what is a carbohydrate? Which is right here. A carbohydrate metabolism is the breakdown of glucose to produce energy. So let's refresh. Glucose is a carbohydrate. It is a sugar. It is a monosaccharide. Glucose is C6H12O6. Glucose is the most common energy source for most bacteria. Glucose is a carbohydrate used by most cells, including human cells. Aerobic respiration and fermentation convert glucose into ATP. So it is possible that bacteria could use carbohydrates such as lactose or sucrose or other sugars, or proteins or lipids to produce ATP. However, most organisms on the planet use glucose to make the majority of their ATP, and they will always choose glucose if it is available. So now we're going to go into the introduction for glycolysis. So glycolysis is the first step in aerobic respiration and in fermentation and in anaerobic respiration. What is glycolysis? It is a 10-step enzyme-driven reaction. We will look at other slides and pictures and animations of this as we proceed through this worksheet. So what does it mean that it's a 10-step enzyme-driven reaction? What that means is it takes 10 steps to go from our initial glucose to our final product, which is called pyruvate or pyruvic acid. Each step requires a different enzyme. If at any of those steps the correct enzyme is not present, then glycolysis will stop. So let's look at the word glycolysis. Glyco means glycogen or stored glucose. Lyse means to split or burst. So in glycolysis, it means the splitting of sugar, in this case glucose. It uses substrate level phosphorylation. Recall from the first video, substrate level phosphorylation is when a microbe or an organism has a phosphate and then donates the phosphate to an ADP, forming ATP. It uses oxidation of glucose to produce pyruvic acid. So let's look at the details. So yes, it is a 10-step driven enzyme reaction. You do not need to know all 10 steps. You need to know the beginning substrate, which is glucose, and the end product, which is pyruvic acid. So let's look at the details. Where does glycolysis occur? In the cytoplasm. The cytoplasm is the jelly-like substance inside the cell. What happens during glycolysis? Again, glycolysis means the splitting of glucose. So in glycolysis, C6H12O6, which is glucose, is split into two 3-carbon molecules called pyruvic acid. For our purposes, we are only going to focus on the carbon. So what does that mean? It means glucose has 6 carbons. We're going to divide those in half. 6 divided by 2 is 3. So pyruvic acid contains 3 carbon molecules. And there's 2 of them because 2 times 3 is 6. So for an example, if someone had 6 apples, which represent the carbons, and they divided those in half, each person would get 3 apples. In this case, we had glucose, which had 6 carbons. We divide them in half, so now each pyruvic acid, which has 3 carbons, is present. So what are the products? 2 pyruvic acids, each of which has 3 carbons, 2 ATP, which were created in those 10 steps, and 2 NADHs. So let's refresh. What is an NADH? NAD plus is a proton carrier. If you see the H present, that means it's carrying protons. So during those 10 steps, electrons and protons were given off, and NAD plus picked them up to become NADH. Those were part of those redox reactions discussed in the first video. In glycolysis, oxygen is not required, so technically we are anaerobic at this point. So we will discuss 3 different types of respiration that occur in bacteria. They are aerobic respiration, which uses oxygen, anaerobic respiration, which does not use oxygen, and fermentation. All 3 of these processes use glucose in glycolysis. We will discuss each of them in the next few slides. Glycolysis. This is step 1 of aerobic respiration, anaerobic respiration, and fermentation. Glycolysis occurs in the cytoplasm. Glycolysis begins with glucose. Glucose, labeled here, is a 6-carbon molecule. During glycolysis, it takes 10 steps to go from glucose to pyruvate or pyruvic acid. You are not required to know any of the 10 steps. You just need to know what goes into the reaction and what the product is. So what goes into the reaction is glucose, and what you get is 2 pyruvates or pyruvic acid. Also during glycolysis, enzymes are used at each of those 10 steps. You produce a total of 4 ATP, but 2 are used for a net gain of 2 ATP. In addition, during glycolysis, 2 NADHs are formed, which are carrying electrons. The electrons released during the chemical reactions in those 10 steps. So this is showing the overview of what happens during glycolysis. Glycolysis, once again, occurs in the cytoplasm. It takes glucose and splits it into 2 pyruvic acids. Everything in this screen you would multiply by 2 because we're going to do the cycle twice. So glucose is broken into 2 pyruvic acids, which forms 2 NADHs and 2 ATPs. So turn in your PowerPoint packet to page 112, where it says Introduction to Aerobic Respiration. And what you're going to see is an equation at the bottom in bold that says Equation for Aerobic Respiration. So we will fill in the top part in a moment. This equation is extremely important and it will be on quizzes or exams. So before we begin, let's kind of break down what this equation is about. The first thing you see on the left, the reactants, is C6H12O6. Recall that is glucose, a sugar or carbohydrate, plus 6O2. O2 is oxygen. So what we're saying is what's put into the bacteria cell's body, so to speak, is food and oxygen. And what's given out, or the products, is 6 carbon dioxide, 6 water, and 38 ATP. Recall when we saw the picture of the Durham tube on several previous slides that the Durham tube allowed us to see whether or not gas was produced. That gas was CO2. So if a bacteria does aerobic respiration, we can see the gas being trapped in that Durham tube. So what we will do as we progress through all of the steps in aerobic respiration is we will see how each of these parts or each of these components of the formula are used in the steps of aerobic respiration. So step 1, which we've already seen, is glycolysis. In glycolysis, glucose, which is there on the left of the equation, is used and broken down into pyruvic acid or pyruvate. During that process, no oxygen was used. So far in this equation, all we have done is use the glucose in glycolysis. The rest of the steps, the prep step, the Kreb cycle, and the electron transport chain will use oxygen, produce carbon dioxide, and eventually produce water and ATP. So here we are on page 113. 113 is an overview of aerobic respiration. Here on the side, we can see the same diagram that we looked at a few minutes ago. This is again the summary of all the steps that take place. We are going to begin with aerobic respiration introduction. What is aerobic respiration? It is the breakdown of glucose and the presence of oxygen. So oxygen is going to be present during this process. It's going to produce the majority of the ATP, or the most ATP, out of any type of metabolism at 30 ATP. It is more than anaerobic and more than fermentation. An example of a microbe that can perform aerobic respiration is Micrococcus luteus. If you recall, Micrococcus luteus is an obligate aerobe when inoculated into fluid thioglycolate. Because it is an obligate aerobe, it has to do or will contain oxygen. Obligate aerobes, such as Micrococcus luteus, can perform aerobic respiration. So we've already discussed glycolysis. Glycolysis was the breakdown of glucose and the presence of oxygen. We will continue to look at the other steps in this process. So after a microbe has taken glucose and broken it down into pyruvic acid, it has one of 3 paths to choose from: aerobic, anaerobic, or fermentation. We are going to focus on aerobic respiration. The second step, which you should know all of the steps in order, is the prep step or intermediate step. The intermediate step or prep step, seen here, is the second step. It is going to occur in the cytoplasm. The third step is the citric acid cycle or Kreb cycle, and the last step is the electron transport chain. Here's our equation for aerobic respiration. This equation is very important in understanding where each of these components came from. So our equation once again is C6H12O6, which is glucose, plus 6 oxygen, yields 6 carbon dioxide, which we can see as a gas, plus 6 water and 38 ATP. So what we've said is we've given our bacteria some glucose and some oxygen. They are going to produce some gas, a little water, and a lot of energy. It requires a lot of energy for those bacteria cells to divide. Again, we're talking about microbial metabolism, which basically equals microbial growth. So now that we are finished with glycolysis, let's move on to step number 2. At the end of glycolysis, we had a 3-carbon molecule called pyruvic acid. Step number 2, the prep step, or intermediate step, occurs in the cytoplasm. Here's what happens. Our 3-carbon molecule, called pyruvic acid, is going to lose a carbon, becoming a 2-carbon molecule. 3 carbons minus 1 carbon gives us 2 carbons. That 2-carbon molecule has a name, which is acetyl. That 2-carbon molecule acetyl then will combine with something called coenzyme A to form acetyl-CoA. So before we continue with acetyl-CoA, let's go back up to the carbons that were lost. So each carbon that was lost from each pyruvic acid combines with oxygen to form carbon dioxide. So pyruvate contained 3 carbons. It lost a carbon. That carbon that was lost combined with oxygen to form CO2. That occurred twice because there were 2 pyruvic acids, giving us carbon dioxide. Again, we can see that occurring in the Durham tube of our glucose broth. Now let's look at our acetyl. Acetyl will combine with coenzyme A to form acetyl-CoA. The purpose of the CoA is to transport the acetyl to our next step, the citric acid cycle. So the end results or products of the prep step are 2 acetyl-CoAs, 2 carbon dioxide, 2 NADHs. So if you recall, NADH's job is to transport electrons and hydrogens, and 0 ATP. One of the ways I like to remember each step is how they're unique from one another. So glycolysis was the only step that would not require oxygen. The prep step is the only step that does not require or produce any ATP. Yes, oxygen is produced, and no, we do not make any ATP. So now we will look at the details of pyruvic acid, or the prep step, on a different slide. On this slide, you can see the prep step in depth, otherwise known as the intermediate step. In the prep step, we have 3 carbons, 1, 2, 3. That was the splitting of glucose. Glucose had 6 carbons, we divided that in half. 6 divided by 2 is 3. In the prep step, the pyruvic acid is going to lose a carbon and become acetyl. Each of those carbons lost become carbon dioxide. So in the prep step, we take pyruvic acid, we lose a carbon to get a 2-carbon molecule. That 2-carbon molecule combines with coenzyme A to form acetyl-CoA. We also do everything twice because we started off with 2 pyruvic acids. So 2 pyruvic acids, 2 carbon dioxide, 2 acetyl-CoA. We also make 2 NADHs. What you're going to notice about this step is we are not making any ATP. This occurs, as you can see over here, in the cytoplasm of our cell. So now let's focus on step 3, the citric acid cycle. The citric acid cycle is sometimes called the citric acid cycle because it produces citric acid. It is also called the Kreb cycle, named after the person that discovered it, Kreb. So where are we in the cell? We are currently in the cytoplasm of the cell. What happens? In the citric acid cycle, our acetyl that we formed in the prep step is going to combine with something called acetyl-CoA. Acetyl-CoA then takes the acetyl to the citric acid cycle. As soon as they arrive at the cycle, they are going to split, and acetyl is going to enter the cycle, and the CoA will go back and pick up another acetyl. So what happens? Acetyl, which is a 2-carbon molecule, is going to combine with something called oxaloacetate, a 4-carbon molecule, to form citric acid, a 6-carbon molecule. This 6-carbon molecule, citric acid, is not glucose. It has different hydrogens and oxygens, which you do not need to know. Then a series of redox reactions occurs. Recall, a redox reaction is oxidation reduction. One molecule receives electrons, the other is going to donate electrons. So what are the products of the citric acid cycle? 4 carbon dioxide, 2 ATP, 6 NADH, and 2 FADH2s. This step or cycle produces the majority of the NADHs and FADH2s. Those will then be used in the last step, the electron transport chain, to make ATP. Oxygen is required for this cycle or step. So now is an up-close and in-depth picture of the citric acid cycle or Kreb cycle. This is step 3. You do not need to know what happens in every step, but let's just go through so we can see what is happening. So here we are at our prep step. And in the prep step, we had our 2 carbon molecule acetyl being formed. Acetyl combined with coenzyme A to form acetyl-CoA. The acetyl then split from the CoA to form citrate or citric acid, a 6 carbon molecule. The cycle then spins in a circle. As the cycle spins, NADH is going to be formed 3 times, so NADH and NADH. And again, the cycle is occurring twice. So 3 NADHs times 2 is 6. Then we have 2 FADH2s, again, 3 times 2. I'm sorry, that one's just times 1. 1 times 2 is 2, so 2 FADH2s. And last but not least, we have ATP of 2, and we have 2 carbon dioxides, and 2 times 2 is 4. To summarize, acetyl enters the cycle. Acetyl is going to form citric acid or citrate. As the cycle spins, we're going to make 6 NADHs, 2 FADH2s, 2 ATP, and 4 carbon dioxide. This cycle does require oxygen and does occur in the cytoplasm of the cell. So before we begin, this is an animation of the Kreb cycle. So the prep step just occurred and the acetyl-CoAs were formed. Also, this animation is of a eukaryotic cell. The concepts are exactly the same. The only difference is every time they mention mitochondria, we're talking cytoplasm. So you should now be on page 113 of the PowerPoint packet. We are to the last step in aerobic respiration called the electron transport chain. It is the last step and is also one of the most important steps because it produces the majority of the ATP. This step is also one of the most confusing stages of the 4. We will finish this page and then we will look at this step in detail and watch an animation. So at the top it says electron transport chain. What is it? A series of redox reactions. So what is occurring is electrons and hydrogens are being passed from one electron carrier to the next. It uses what we call chemiosmosis to produce or generate ATP. We will discuss chemiosmosis on the bottom of this worksheet. Where does this occur? Well, we need to create what's called a concentration gradient, meaning a difference on the amount, in this case protons, on one side of a membrane than the other. So we were going to use the cell membrane to create that concentration gradient of prokaryotic cells. In a eukaryotic cell, this would occur in the mitochondria, but prokaryotic cells do not contain mitochondria. So let's start with classes of carrier molecules. So what is a carrier molecule? A carrier molecule is a molecule that carries a substance, in this case, electrons and protons. There are 4 classes that we will discuss. NADH dehydrogenase, which is an enzyme, it ends in ase, and it removes electrons from NADH and makes it become back NAD+. The second, flavoproteins. They contain flavin, think riboflavin, and an example is something called FMN. Next, coenzyme Q or ubiquinones, which are the same structure. They transfer electrons to cytochromes, which are small non-protein carriers. Lastly, cytochromes are proteins with iron containing groups. Examples include cytochromes B, C, and A. So the best way to think about what these carriers are, it is as if you were in a relay race and all 4 members of the relay race were positioned one after the other. They would pass, in this case, the baton from one person to the next. In this story, instead of passing a baton, we will be passing electrons from one electron carrier to the next. And as we do that, protons or hydrogens are being pumped to the outer membrane and creating a concentration gradient. We are going to now look at the steps in the electron transport chain. There are 7 steps, and you should know these in order. Step 1. NADH dehydrogenase removes electrons and protons or H+, from NADH and transfer them to FMN. FMN then becomes FMNH2. That is a redox reaction. So where did the NADH come from? Recall, glycolysis produced 2 NADH. The prep step produced 2 NADH. The citric acid cycle produced 6 NADH. So each of those NADHs is going to enter the inside of that cytoplasm and be stripped of its protons and passed along the chain. Next, FMNH2, which was just produced in step 1, transfers its electrons to coenzyme Q. Again, this is a baton, kind of a relay race. We're just passing them from one to the next. From coenzyme Q, they are transferred to cytochromes, and from cytochromes, we're transferring the electrons to oxygen. We are discussing aerobic respiration. So what is called the final electron acceptor, the end of the line, is oxygen. Oxygen and protons, hydrogens, then are going to form water inside the cell, which we will explore more on the next slide. The hydrogens build up outside of the membrane and a concentration gradient forms. This is called chemiosmosis. Hydrogens diffuse into the cell, and ATP synthase, an enzyme, catalyzes the reaction of ADP forming or combining with another phosphate to form ATP. So how does all this work? Chemiosmosis and ATP production. So in this process, chemiosmosis occurs. So you have a general understanding of osmosis. Osmosis was the diffusion of water across a cell membrane. So diffusion means the movement from high to low, obviously water, and it was a cell membrane. So in chemiosmosis, it is the diffusion of protons across the cell membrane. So it says, mechanism that uses a proton gradient across the cell membrane to generate ATP, using the enzyme ATP synthase. So as those hydrogens move back into the cell, or back into the cytoplasm, ADP plus P are going to use that energy force to form ATP. So what are the products? 6 water, 34 ATP. And is oxygen required? Yes. Oxygen is required to continue that diffusion of hydrogen to make that ATP, which we will see on the next slide. So here we are at the last step, the electron transport chain. This is the same image or picture that you have on page 113. This has just been enlarged so you can see it a little bit better. So before we do anything else, let's get our bearings of where we are. So here we are inside a cell. So this is a prokaryotic cell, and we are initially in the cytoplasm. And it does say down here, cytoplasm of prokaryote or mitochondrial matrix of eukaryote, but we're going to focus on prokaryotes. So what we're going to be doing is moving or shuttling right here. This is our cell membrane, electrons and protons from one side to the other. You can also see here up at the top, you see NADH becoming NAD, which is then passing electrons to FADH2, to FAD. We see our FMN, our cytochromes, our coenzyme Q. As we're passing these things down, our final electron in this system, or this chain, is oxygen, our final electron acceptor. And this is a redox reaction. So just to go back over that a little bit, this was the passing of the baton idea. So NADH is going to pass them to FMN, and that's going to happen by NADH dehydrogenase. FMN passes the protons and electrons to coenzyme Q. Coenzyme Q to all of the cytochromes, which you do not need to know in order, to oxygen, and last we're going to form water. So let's go down here in this blown up picture of what's actually happening. Here's our cell membrane where we're going to use for chemiosmosis. So here was our NADH, and it's going to become NAD+. That's a redox reaction. NADH dehydrogenase is the enzyme responsible for that. That's our very first electron carrier. Next, we're going to pass them meaning electrons, to FMN, to coenzyme Q, so it's the same picture just blown up bigger, to the cytochromes, more cytochromes, and eventually to oxygen. So while that's happening, all these protons are being shuttled into this outer space between the cell membrane and the cell wall. This creates what we call a concentration gradient. What that means is we have more hydrogen on the outside than on the inside. They are going to want to diffuse. Osmosis is diffusion. So they're going to diffuse from high to low. They're going to diffuse from high out here, back in to low on the inside. As they do that, I kind of, using the example, this is like a turbine. Sitting right here is ADP plus P. So as that happens, as those hydrogen diffuse back in, they're going to combine with oxygen to form water. And as they do that, they're going to form ATP. ATP synthase is the enzyme responsible for that formation. Now let's kind of discuss what's happening with this oxygen. We call oxygen the final electron acceptor because it's the end of the line. If the oxygen was not present, the hydrogens would stop diffusing. They would each reach equilibrium. So once they reach equilibrium, they would stop making ATP and the cell would eventually die. So let's look at an animation of how this works.